Whenever somebody mentions gas in the body, what comes to our mind is a very useless and wasteful entity, that is gas in the abdomen, the entity that needs to be expelled out of the body as a waste. But can a gas have an important physiological role to play in the body and as important as a neurotransmitter? Yes, it does happen in fact. And this video is on gases acting as neurotransmitters. We have two gases which definitely are known to act as neurotransmitter in the brain or in the CNS in general, in the nervous system. They are nitric oxide and carbon monoxide. Let's understand more about these gases as neurotransmitters. This is the newest category of transmitters. What is so special, unique about these neurotransmitters? They are neither packaged inside the vesicles and therefore nor released by exocytosis. Look, when we say a conventional neurotransmitter, we are aware about the low molecular weight transmitters like acetylcholine or noradrenaline, or high molecular weight transmitters, the neuropeptides like neuropeptide Y, etc. This is a third category, different from these two. How is it different? Look, normal conventional neurotransmitters, they are packaged in the vesicle, and whenever the impulse arrives, the vesicle will discharge the neurotransmitter by a process of exocytosis. Gas neurotransmitters are not packaged inside the vesicle in the presynaptic terminal. It is free in the presynaptic terminal. That means it will simply diffuse out of that neuron and will act on the next neuron, not packaged in the vesicle nor released by exocytosis. All right, you will see some more amazing features of these gas neurotransmitters. Synthesis is triggered by depolarization of the nerve terminals. Okay, this is like on demand. Whenever the impulse arrives, the gas neurotransmitter is synthesized then and there, and then it will be released on demand. Okay, so when impulse arrives at the nerve terminal, there is influx of calcium. In the conventional neurotransmitters, influx of calcium is required for exocytosis, but not here. Here the calcium comes in for what? It is for activation of enzymes involved in the synthesis of gas neurotransmitter. Yes, so calcium influx occurs, activation of enzymes involved in the synthesis of the gas neurotransmitters. So neurotransmitters will be synthesized then and there on demand and then will be released. Take some more. No specific receptors to bind. Conventional neurotransmitters, they are on the postsynaptic neuronal membrane, and when the neurotransmitter is released, it combines with those receptors. In the case of gas neurotransmitters, there are no specific receptors on the postsynaptic membrane. Then how does it act? It acts by actually triggering the guanylyl cyclase enzyme activity, which means there will be formation of cyclic GMP in the next neuron, and that will exert the effect of the gas neurotransmitter. So very unique properties, very different from our conventional wisdom about the neurotransmitters. Okay, some more. No reuptake or enzymatic degradation. You know, some neurotransmitters like glutamate, there is a reuptake of those transmitters by the presynaptic neuron once they have started their action. Some of them are enzymatically degraded like acetylcholine. In the case of gas neurotransmitters, what happens is they simply diffuse out of the synaptic cleft, or they get bound to superoxide anions. That is how their action will be terminated. Now with this background, let's see those two gas neurotransmitters, the NO and CO. Nitric oxide mainly secreted in those areas of the brain responsible for long-term behavior. So long-term memory, which involves long-term potentiation in hippocampus, this is where NO is said to be involved. Also by ENS neurons, this is already known to us. You know, ENS neurons have two most common inhibitory transmitters, VIP and NO. So this is already known. How NO is different compared to low molecular weight transmitters. So take some more unique properties. It appears to be released by both presynaptic and postsynaptic neurons. Yes, very unique, very special. Our conventional neurotransmitters like acetylcholine, etc., they are released by one neuron and they will act on another neuron, next neuron. And therefore, we call them as presynaptic and postsynaptic neurons, isn't it? Gases can be released by both these neurons, so-called presynaptic and postsynaptic. Now we can't call them pre and postsynaptic because both neurons can release gas on each other. So it's a synapse, but without calling them pre and postsynaptic neurons. In fact, there is one more property which is recently discovered. This is again fascinating. In our conventional neurotransmitters, they are released by one neuron and they will act on the immediate next neuron, right? That's it. In the case of gaseous neurotransmitters, they are released by one neuron. They will not only act on the next neuron, but they can diffuse throughout that neuron, travel, and can act on third neuron as well, next neuron. This is only possible with a gaseous neurotransmitter. Isn't it amazing? All right. So that's about the special feature about the NO. It is not preformed and stored in the vesicles. We already saw that. It is synthesized almost instantly, on demand, wherever needed and whenever needed. Calcium influx is not for its release, but for its synthesis. Yes, we saw all that. It diffuses out of the presynaptic terminal and not in the vesicular quanta. We saw that already. Now this is again very unique and special. It does not greatly alter the membrane potential of the postsynaptic membrane. In the case of other neurotransmitters, when they are released, they generate membrane potentials on the next neuron, EPSPs and IPSPs. So altering the membrane potential is the core action by the neurotransmitters, but not in these gas neurotransmitters. They don't generate EPSPs and IPSPs. Rather, the NO changes the metabolic functions of the postsynaptic neuron, and which then modifies the neuronal excitability for prolonged periods. Yes, that's how they act, without generating the EPSPs and IPSPs, without greatly altering the membrane potentials of the next neurons. All right, very unique, very special. Let's also see carbon monoxide as the neurotransmitter. We would have thought carbon monoxide is a poisonous gas. Yes, it is. Inhaled in large quantities for prolonged durations. Yes, it is. But carbon monoxide is also endogenously formed. Yes. Look, normally all our metabolic reactions as an end product, they generate carbon dioxide. But there is only one reaction in the body that generates carbon monoxide. And what is that reaction? Reaction of degradation of heme. Degradation of heme generates carbon monoxide. So formed by enzymatic degradation of heme by heme oxygenase 2. Therefore, carbon monoxide can be endogenously generated. Please keep this in mind. Opening up of the porphyrin ring, or rather a degradation of heme and opening of porphyrin ring. This will release carbon monoxide. Okay, it's a diffusible gas which acts as a neurotransmitter, endogenously formed in the CNS and ENS, enteric nervous system neurons. And as I mentioned, the only reaction in the body that generates carbon monoxide rather than carbon dioxide. Okay, it activates guanylyl cyclase. Yes, we know about that now. Gas neurotransmitters, they activate guanylyl cyclase and therefore they increase the cyclic GMP levels in the target neurons or postsynaptic neurons. Okay, it may have a role in neurotransmission of olfaction. What research has told us so far is that this neurotransmitter, carbon monoxide, may have a role in olfactory sense. then nociception, pain, and long-term potentiation in hippocampus. Did you note one thing? Long-term potentiation in hippocampus, that's the basis for long-term memory. NO and CO both have a probable role to play. So gas neurotransmitters, very important for long-term memory. You can remember like that. And it has possible role in attenuation of arginine vasopressin release. It's an amazing, amazing physiology, and physiology as a subject overall is really amazing subject.